Our first question is, how is the structure of eukaryotic and prokaryotic cells different? Our second question is, how is the organization of prokaryotic and eukaryotic genomes different? Chapter 2 is about reproduction and cell division. How prokaryotes and eukaryotes reproduce is largely determined by the structure of their cells and by the organization of their genomes. So we'll start this video by comparing eukaryotes and prokaryotes. Prokaryotes are relatively simple, single-celled creatures. There are two domains of prokaryotes, bacteria and archaea, but we'll mainly deal with the bacteria. A word on grammar here. A bacterium is a single cell. Bacteria is plural. Some bacteria cause human disease, but most are non-pathogenic, free-living organisms. Eukaryotes are more complex and can be single-celled, like this euglena, or multicellular, like humans and plants. Prokaryotes have a relatively simple cell structure. Going from the outside in, some bacteria have a capsule, which provides protection and, for some species, helps them infect their host. Nearly all prokaryotes have a cell wall, and inside that is the cell membrane that keeps the interior of the cell separate from the environment. Inside this semi-permeable bag is the cytoplasm, which contains everything within the cell, including the many proteins that are carrying out cellular functions. There are also many, many ribosomes, which produce those proteins. Eukaryotes are much more complex. Eukaryotic plant cells have cell walls, but animal cells don't, while both plant and animal cells have cell membranes. Unlike prokaryotic cells, eukaryotic cells have organelles. These are compartments within the cytoplasm that are surrounded by a membrane and carry out various cellular processes. There are many kinds of organelles, including the vacuole, Golgi complex, and the mitochondria. The mitochondria basically burn sugars with oxygen to yield energy. Plants also have chloroplasts, where they carry out photosynthesis, which is essentially the production of sugar by splitting water molecules using energy from sunlight. Eukaryotes also have ribosomes, which, as in prokaryotes, make proteins that carry out cellular functions. Another important difference between eukaryotes and prokaryotes is size. Prokaryotic cells are typically many times smaller than eukaryotic cells. You could fit millions of bacteria into the space taken up by this tiny dot. For a super cool way of viewing the size of cells and their molecules, check out the link below this video. And this is a good time to point out that when I direct you to a website like this one in one of my videos, you should consider that website part of this course. Often, you'll have to answer questions about it, so make sure to visit the site. For this website, there's no need to memorize all the information there. I just want you to get an idea of the relative sizes of cells, their organelles, and their molecules. Okay, so here, we'll look at the differences in how the genomes of prokaryotes and eukaryotes are organized. The vast majority of DNA in prokaryotes is in one chromosome. In the cell, this DNA is coiled, but if we uncoil it and look at it under a microscope, we see that its shape is circular. Smaller chromosomes called plasmids, sometimes present in many copies, may also be present. This smaller DNA molecule in the micrograph is a plasmid. While prokaryotes have one circular chromosome, eukaryotes have one or more chromosomes, often many of them, which are also coiled. Eukaryotic chromosomes are linear, that is to say that each chromosome has two ends. Prokaryotes only have one copy of their main chromosome, so they only have one copy of each of the genes that are on that chromosome. Therefore, the genotype of most prokaryotic genes is written with only one allele, such as FLG+. They are therefore haploid for these genes. This is one reason it's easier to study gene function in prokaryotes. You only have to mutate one copy of the gene to carry out mutant analysis. This has been done for many genes in the model organism E. coli, so that we know the function of many genes in this bacterium. For genes present on plasmids, these can be present in multiple copies. For example, if this cell has two plasmids with the LAC-Z gene on them, they have 3 copies of that gene total, one on the chromosome and one on each plasmid. Any genotype of this gene will have to include what alleles the chromosome and these 2 plasmids carry. In eukaryotes, the chromosomes a species has is known as its karyotype. And a picture of a karyotype is a karyogram, like this human karyogram. As we can see, humans, like most eukaryotes, have 2 of each chromosome. For chromosome 11, here's 1 copy, and here's the second. Since most eukaryotes generally have 2 copies of each chromosome, they have 2 copies of each gene. This is why eukaryotic genotypes generally have 2 alleles, like the genotype HBB+, HBB-. They are, therefore, diploid. However, not all eukaryotes are diploid. As we'll see later, some are polyploid, or have more than 2 copies of each chromosome. Additionally, many eukaryotes spend some part of their life cycle as non-diploid organisms, as we'll see on the next page. Here we see the life cycles of humans, plants, and fungi. Eukaryotes, like humans, do make haploid cells in the form of sperm and egg, which fuse during fertilization to form the diploid zygote. In plants, the products of meiosis are haploid spores, which then divide to form a haploid gametophyte, which produces the haploid sperm and egg. In fungi, most of the life cycle is haploid, with the zygospore being diploid. As you've seen, haploid cells have 1 of each chromosome, diploid cells have 2 of each chromosome, but some species, especially plants, have more than 2 copies of each chromosome and, therefore, have more than 2 copies of each gene. Having more than 2 copies of each chromosome is called polyploidy. The prefix poly means many. In this case, many or more than 2 chromosomes of each type. The type of polyploidy being shown here is tetraploidy, because we see 4 copies of each chromosome, the prefix tetra meaning 4. A species with 6 copies of each chromosome is hexaploid, a species with 8 copies of each chromosome is octaploid, and so on and so forth. Because of polyploidy and because there are haploid parts of eukaryotic life cycles, it's a bit of an overgeneralization to say that eukaryotes are diploid. It's probably more precise to say that most multicellular eukaryotes are diploid most of the time. Let's see some other differences in genome organization between prokaryotes and eukaryotes. In prokaryotes, the region where the chromosome is found is called the nucleoid region, and it is not separated from the rest of the cytoplasm. So the nucleoid is not an organelle. It's just the part of the cytoplasm where the chromosome is found. Eukaryotic chromosomes are housed within a membrane-bound nucleus. This has important consequences for how the cells divide, as we'll see later. Mitochondria also have multiple copies of their own circular DNA, as do chloroplasts in plant cells. When the cell divides, these organelles must also replicate themselves and their DNA. Prokaryotic genomes are relatively small, but can range quite a bit in how many genes they have. Parasitic prokaryotes can have down to just a few hundred genes, whereas more complex free-living prokaryotes typically have a few thousand genes. Eukaryotic genome size also varies wildly and can range from 2,000 to 30,000 genes. The 23 human chromosomes are collectively about 3 billion nucleotides long and have around 22,000 protein-coding genes. Yeast, a much simpler eukaryote, has 16 chromosomes and about 6,000 genes. Prokaryotic DNA can fit inside the cell in a relatively unpacked state. Because eukaryotes have so much DNA, 6 feet long in human cells, if it was stretched out end to end, it has to be packaged in order to fit into the nucleus. Check out this video from HHMI to see how this is accomplished.
In this animation, we'll see the remarkable way our DNA is tightly packed up to fit into the nucleus of every cell. The process starts with assembly of a nucleosome, which is formed when 8 separate histone protein subunits attach to the DNA molecule. The combined tight loop of DNA and protein is the nucleosome. Multiple nucleosomes are coiled together, and these then stack on top of each other. The end result is a fiber of packed nucleosomes. This fiber, which at this point is condensed to a thickness of 30 nanometers, is then looped and further packaged using other proteins which are not shown here. This remarkable multiple folding allows 6 feet of DNA to fit into the nucleus of each cell in our body, an object so small that 10,000 nuclei could fit on the tip of a needle. The end result is that the DNA is tightly packed into the familiar structures we can see through a microscope, chromosomes. It is important to realize that chromosomes are not always present. They form only when cells are dividing. At other times, as we can see here at the end of cell division, our DNA becomes less highly organized.
This structure that you saw in the video contains both DNA, histone proteins, and other scaffolding proteins not shown, and is known as chromatin. Which is what chromosomes are made of. The double helix of DNA can be greatly condensed into more tightly packed forms of chromatin. Double-stranded DNA is wrapped around 8 histones, forming a nucleosome. And if an additional histone, H1, is added, the chromatin condenses further into 30 nanometer fiber. If proteins called scaffold proteins are added, the DNA adopts a progressively more condensed form, culminating in the metaphase chromosome, which forms during cell division and is also what we see in karyograms. Keep in mind, though, that chromosome packaging, also called chromosome condensation, is reversible. Chromosomes like this only appear when the cell is dividing. Why does the cell only finish condensing the chromosome during cell division? Chromosomes are condensed so that they can be moved and evenly distributed to the daughter cells. It's the same reason you pack your clothes before taking a trip instead of carrying them on hangers. It's just easier to carry them in a condensed form in your suitcase. When a cell is not dividing, the chromosomes are decondensed, and their DNA is less tightly associated with histones and scaffolding proteins. Because of this, the DNA is spread around the nucleus. This is a picture of uncondensed chromosomes from an actual cell, where each chromosome has a different color. Chromosomes are decondensed, so the proteins necessary for transcription can access the DNA and transcribe the genes on the chromosome.